Welcome back, everyone, and we are going to continue our discussion on GI physiology. Today, we're going to talk about motility and we're going to talk about secretion. And we'll touch on the beginning of digestion. These topics can be a little bit heavy at times, so it'll be worth your while if we're talking about a process to make sure that you go through that process a couple of times to make sure that you understand all the steps and why all the steps are happening. So when we talk about motility in the GI tract, it really serves two major purposes. The first is it is for moving food throughout the GI tract. And the second one is that it helps mechanically mix the food into uniformly small particles. Mixing actually helps maximize the surface area that comes into contact with the small particles, and therefore will have the maximum amount of enzymatic reaction that can happen to break down the foodstuff further. So motility is determined by a couple of things. First, there has to be some sort of an input from nerves or input from specific hormones or those paracrine signals that we talked about that go from one cell type to a cell type that's in the vicinity. Without input, there's really nothing to tell the GI tract to contract and to have things move. The second thing that you need is muscle action in order to help things move along. And the specific type of muscle is smooth muscle. This is the type of muscle that is not under voluntary control. And we're going to talk about the specific mechanism by which smooth muscle contracts. But first, we're going to talk about the three general types of muscle contraction that occur in the GI tract. And these three types are referred to as the migrating motor complex, peristalsis, which probably most of you have heard of, and segmental contractions. The migrating motor complex is the type of movement that occurs in the GI tract between meals. So it's not while you're eating a meal, it's not just after a meal, it's in the period of time when you're not eating. And it helps food move along from the stomach to the large intestine. Contractions will generally start in an empty stomach, and then it moves from section to section in sort of a migratory pattern. So it starts in the stomach, and then it migrates to the small intestine, then the movement migrates further through the small intestine, and eventually migrates through the colon. Really serves a housekeeping function, keeping things clean in the GI tract. A full cycle from the beginning, which is in the stomach, to the very end, which is the end of the large intestine, takes approximately an hour and a half. There are two types of contraction that happen following meals, and that's immediately following meals. And so these two patterns are peristalsis and segmental contractions. Segmental contractions actually occur after a meal, but peristalsis, sort of like peristalsis that occurs while you're eating as well. So peristalsis is like a wave, and so it moves from one region to the next fairly quickly to move a bolus of food from one segment of the GI tract to another segment of the GI tract. And so it's this progressive wave of contraction. It's different from the migrating motor complex in that it moves a bolus of ingested material after you've actually eaten it. It's not a housekeeping function at all. It's helping things move along. So the way that it works is at time zero, there is contraction of the circular muscles just behind the bolus of food. So if you remember what the circular muscles are, when those contract, the luminal diameter decreases. And so you can see a decreased luminal diameter here compared to the distal segment. And this contraction of the circular muscles pushes the bolus of food forward to what is referred to as the receiving segment. And then the next thing that happens is that the circular muscles in the receiving segment will contract and push the bolus of food along to the next receiving segment. Peristalsis is fairly quick. It occurs at a rate of anywhere between 2 and 25 centimeters per second. The major function of peristalsis is to move food forward in the GI tract. It contributes a little bit to mixing within the stomach, but the major feature of peristalsis is that it functions to move food forward. It is controlled by hormones, controlled by those paracrine signals, controlled by autonomic nervous system influences. We're going to talk about what some of those hormones are, what some of those paracrine signals are, a little bit later when we talk about the regulation of GI function. Segmental contractions are different from the other two in that it's just short regions of the intestine, and mostly the small intestine. And these short regions contract and relax in an alternating pattern. And it actually makes use of both the circular muscles and the longitudinal muscles. So the circular muscles, when they are contracted, the longitudinal muscles are relaxed. And then you can imagine when they flip, when the longitudinal muscles are contracted and the circular muscles are relaxed. And this back and forth movement, or this back and forth contraction, doesn't really move anything along the GI tract. Its major function is to churn the intestinal contents. And this helps ensure that the maximum amount of foodstuff is in contact with the most amount of epithelium so that digestion can happen and absorption can happen. And so here's what segmental contractions would be like. So these are different colors just to depict that this is the region in which different parts of partially digested food are. So there's the yellow region, the green region, the purple, and the blue. And you can see that the longitudinal muscles, when they contract, they shorten this part of the intestine. And when the circular muscles contract, it decreases the luminal diameter. And then this pattern switches to now the longitudinal muscle in that region is now relaxed, and the circular muscle instead is contracted. And what this helps do is it helps to mix up all the contents to ensure that the maximum amount of surface area is in contact with the maximum amount of partially digested foodstuff. And it maximizes digestion, maximizes absorption. So if you want to differentiate the types of motility within the GI tract, the migrating motor complex, or the MMC, is the major housekeeping function, and it's forward movement between meals, specifically between. Peristalsis is like a wave, and it's forward movement of foodstuff just after a meal. And segmental contractions, there's no net movement, it's just mixing and churning of all the contents. So the big question is, how does this muscular contraction occur? And that's via smooth muscle. The way that smooth muscle is organized are in these smooth muscle cells. So this is cell 1, and this is cell 2. You'll notice that there's a few things within this cell. There's all these dark regions, these dark blue regions. These are called dense bodies. And this, if you notice, will connect some of the muscle filaments. And there's really two major components that are responsible for contraction. Those two major proteins or components are actin and myosin. And so if you've learned about muscle contraction in the past, these two words should not be new to you. But the thicker filament, or this is the blue region here. Also, you can see the blue region in the center here. These are called myosin filaments. And what you'll notice is that there's these head regions that are kind of hinged, and they're connected to this red filament. And this red filament is quite a bit thinner than the blue filament or the myosin filament. And these thinner filaments are referred to as actin. They're actin filaments. And so when muscle contracts, no matter where the muscle is, what ends up happening is that these myosin heads attach onto the actin and the heads swivel forward. And what it does is it pulls the red filaments closer together. And in essence, what that will do is it will shorten the entire muscle cell. And that would be a contracted state. When it relaxes, those myosin heads detach from the actin filaments. And when it relaxes, the muscle cell lengthens again or relaxes. And so the way that it's really connected is that there, between each cell, is some connective tissue that ensures that there's some linking of the smooth muscle cells, so that the force from one muscle cell contracting can be transmitted to the adjacent cells. And the adjacent cells will also have the same level of contraction. Now, when myosin, this thicker filament has those heads along the entire length that can attach to the actin. And when the myosin is active, it will attach to the actin fibers and it will pull them closer together, resulting in muscle contraction. It is absolutely essential that there is calcium present when this happens. So you need the ion calcium. And there's a whole molecular process by which smooth muscle contraction occurs. If you've learned about muscle contraction in the past, and you've talked about troponin and tropomyosin and all that stuff, that's not the same mechanism of muscle contraction here. We're talking about a completely different type of muscle contraction. This is smooth muscle contraction, which uses a different type of approach. And we're going to go through it step by step. So I'm going to just orient you here. What we have here is this darker purple area. This is the smooth muscle cell membrane. The opening here is a calcium channel to allow calcium to come in. The lighter purple is the outside of the cell, so the extracellular fluid component. And then the yellowish area and everything inside of the yellowish area is the inside of the cell, or the intracellular compartment. There is this thing called the sarcoplasmic reticulum. This is a very important storage organelle for calcium within inside muscle cells. It's essentially very similar to an endoplasmic reticulum. It's a smooth endoplasmic reticulum, and that doesn't contain ribosomes, if you're interested to know that. So it stores calcium. And so the first thing that happens in a case where you want smooth muscle to contract is that there's going to be some sort of a signal come across the cell to tell it to contract. Whether there's a nerve impulse that comes and tells the muscle cell that it needs to contract, or there's a hormone that binds, something is going to happen whereby calcium is going to be allowed to enter the cell. And that's the first step. You have to have calcium in order for muscle contraction to occur. And so when this calcium enters the cell, the relative amount of calcium on the inside of the cell increases. So you get an intracellular calcium concentration increase. Calcium at the same time is released from this sarcoplasmic reticulum. And the function of that is to, again, increase intracellular calcium concentrations. After this calcium enters the cell, it binds with a very specific molecule. And this particular molecule is a specific protein called calmodulin. Probably never heard about calmodulin before, but it's very, very similar to troponin. If you know about the other type of muscle contraction, and you've heard about troponin, where calcium binds to troponin, calmodulin has a very similar molecular structure to troponin, and that's why calcium binds to it. It's just a different molecule. And when this calcium binds, it actually makes something called a calcium calmodulin complex. That's what we call that. Calcium calmodulin complex. So calcium is bound to this calmodulin. And this actually acts as a molecule that can activate an enzyme. And the specific enzyme that this calcium calmodulin activates is this thing called MLCK. So when in the presence of calmodulin and calcium, inactive MLCK, or myosin light chain kinase, will become activated. And if you remember from our talks about enzymes, a kinase enzyme is very important in phosphorylation. So whenever you see that word kinase, which we see here, there's a phosphate added somewhere. So we have active myosin light chain kinase. And when it's activated, what this myosin light chain kinase does is, just like I told you, it phosphorylates something. It actually adds a phosphate group to the heads of myosin. And so it uses ATP to do that. So it helps hydrolyze or break down ATP into ADP plus this phosphate. And the phosphate is bound to the head on myosin. Now, that phosphate, when it's added to the head of myosin, it allows for the myosin heads to bind strongly with actin. And when it binds strongly with actin, this head will swivel forward and it'll pull the actin filaments together. And what you end up with is increased muscle tension and shortening of the muscle cell. So you get contraction. That's the process of muscle contraction. I'll go through it again sort of quickly as a little review. Calcium enters the cell from both the extracellular fluid and from the sarcoplasmic reticulum. That calcium will bind to that protein calmodulin. Together it makes a calcium calmodulin complex. It activates myosin light chain kinase, and that myosin light chain kinase causes ATP to be hydrolyzed. The ATP is converted into ADP plus that phosphate. That phosphate is bound to the myosin head, and the myosin head actively binds to the actin. And when it does, those myosin heads swivel forward and increase the amount of muscle tension that is within the cell, and it shortens the cell. So go through that a few times. Make sure that you really understand the process of smooth muscle contraction, because there's also the process of smooth muscle relaxation, which is now we got to go in the reverse order to make sure that that muscle cell can be brought back to its normal position and its normal size. So smooth muscle relaxation starts with the calcium in the cytoplasm, or the cytosol, is actively pumped out of the cell. So there's a calcium pump. That calcium pump is of a calcium using ATP, and it pumps the calcium out. It pumps the calcium back into the sarcoplasmic reticulum. And effectively what you have is a reduction in the intracellular calcium concentration. That's not good for muscle contraction, because you need intracellular calcium in order to have muscle contraction. But that low calcium on the inside of the cell allows for the calcium calmodulin complex to dissociate. And so calcium will unbind from the calmodulin because there's less calcium on the inside of the cell. And think of it like a chemical reaction. Calcium calmodulin complex yields calcium plus calmodulin. If that calcium level goes down, it's going to shift the equilibrium so that calcium calmodulin complex breaks down more. And so more calcium can be pumped out. And then the calmodulin is ready for the next time that intracellular calcium increases. The next thing that has to happen is that you have to remove that phosphate from the myosin head. And that's done by an enzyme called myosin phosphatase. Just like a kinase adds a phosphate, anytime you see an enzyme that ends with phosphatase, it means that a phosphate is removed from a molecule. And so myosin phosphatase is exactly what its name implies. It's removing a phosphate from the myosin head. So the myosin head, that phosphate gets removed. And with less phosphate bound to the myosin head, you get decreased binding of the myosin head to the actin filament. And with decreased myosin head binding to the actin filament, you get decreased muscle tension. So there's no longer contact between the myosin head and the actin filament, and so the muscle relaxes. So make sure you really understand that process. It's an extremely important one to know. It's going to come back in our respiratory section when we talk about bronchial smooth muscle contraction. It's the exact same process. Any smooth muscle contraction you have in your body, same process to this. We'll talk a little bit about secretion. So there's a lot of things that are secreted in the GI tract. About 9 liters pass through the GI tract every day. We already know that 7 liters of this is from secretions, and about 2 liters comes into the GI tract from the mouth, from the external environment. When we look at these 7 liters that are secreted, they can be split into 2 parts. Half of it actually comes from the accessory organs. So the gallbladder, the liver, the pancreas, and the salivary glands. And then 3.5 liters is actually from the digestive tract itself, from the stomach, the small intestine. Those are 2 of the major organs that would contribute to that 3.5 liters from the digestive tract. And there's lots of secretions. There are protons that are secreted, or acid. There's bicarbonate ions that are secreted. There's mucus, there's bile, there's digestive enzymes. There are a lot of things that are secreted. We're going to talk about a few of them. The major ones that we are going to focus on in this course are the secretions of the stomach. These are going to come back to bug you a lot in this course. I really want you to know every single one of these. It's very important that you know all these. So I'm going to leave it with you to go through all of these types of cells, make sure that you know all these types of cells, make sure you know what they secrete, because you're going to have to know all of them. The stimulus for secretion or for release, I'll tell you what it is. I'll go through that with you in a little bit more detail in the last section of this of the GI unit. And I want you to know the function of all of these things as well. It seems like a lot, but it's not. They're very well connected to each other. The ones that are going to become, actually the ones that are most important are all of them. And you're going to see there's a lot of interplay between these last 5, lots of interplay. So when we talk about GI regulation and the phases of digestion, this becomes very, very important. Okay, so one of the secretions that we're going to talk about in a lot of depth here is acid secretion. So the stomach makes hydrochloric acid. And so it's within the gastric glands itself that there are cells that produce hydrochloric acid. And these specific cells are called the parietal cells. Makes between 1 and 3 liters a day, depending on the activity within the stomach. Interestingly enough, the pH could get very low. It's around 2 in the majority of areas within the stomach, but it can get as low as 1 in the deep valleys of the gastric glands. So that's a pretty acidic pH. And here's how protons are secreted in the lumen of the stomach. So we're going to orient you here. So there's a blood vessel, capillary. There's this orange cell is a parietal cell. And then this blue region to the left is the lumen of the stomach. So the first thing that has to happen in the secretion of protons is that you actually have to have a water molecule within the cell, and there's lots of water within the cell. All your cells have water to some degree. The first thing that has to happen is that the water molecule has to dissociate, and it dissociates into two things. Number one, it dissociates into a hydrogen ion or a proton. This is the acid. And it dissociates into a hydroxide ion or OH negative ion. That proton is what we want secreted into the lumen of the stomach. And so we can see that this proton gets pumped out by primary active transport. It gets pumped out in exchange for a potassium ion. And so this particular pump that does this job is called the proton potassium, or the H plus K plus ATPase. Remember, ATPase always signifies primary active transport. Well, we're not done yet. There's a reason for that. This hydroxide ion is extremely toxic to the cell. It can't stay like that. Hydroxide ions are extremely alkaline, and if you increase the alkalinity within a cell, the cell will die. So you got to get rid of that hydroxide ion. So the way that we do that is that we combine the hydroxide ion with carbon dioxide. You might be asking the question, where did carbon dioxide come from? Water's in the cell, I get it, but where did carbon dioxide come from? Carbon dioxide came from cellular respiration. Your cells are constantly making CO2 by breaking down glucose and by oxidizing some of the biomolecules for energy. So it's not out of the question that you're just going to have carbon dioxide sort of appear out of nowhere or seemingly out of nowhere in a physiologic reaction. And so this particular process occurs in the presence of an enzyme. You'll notice that there's this written in blue as Ca. That Ca stands for carbonic anhydrase. I should actually write that. I'm going to just exit that here, and I'm going to write it in the slide. Write it in the slide. Presence of carbonic anhydrase. Just so that you have it in your notes as well. Go back to presentation view. Okay. So now we've essentially got rid of that hydroxide ion. We're now left with a bicarbonate ion, which also we don't want too much bicarbonate in the cell either. So we get rid of that. We get rid of that bicarbonate ion. And so that bicarbonate ion gets gets transported out of the cell. And it's in exchange for chloride, a chloride anion. That chloride anion travels through the parietal cell and goes through open channels on the luminal side of the cell and enters the stomach as well. So essentially what you have is hydrochloric acid, the H plus and the Cl minus. One thing I want you to notice about the processes that used transporters, whether it's a primary active transporter at step 2, or whether it's a facilitated diffusion or type process that occurs at step 4. I want you to notice the charges on the substances that were moved. H plus has a charge of +1. K plus has a charge of +1. H plus went out of the cell, K plus came into the cell. After that process happens, there's no net change in the charge of the cell. the cell stays exactly the same charge. You lose one positive charge, you're gaining another one. It's very important for the cell to maintain this sort of neutrality. You don't want there to be a buildup of charge on one side of the cell over another. Unless you're a nerve cell and that's the way that you communicate, or unless you're trying to build up a charge within the cell so that certain transporters can work. But in general, these transporters are going to try to maintain electrical neutrality. Same thing happened at step 4. Bicarbonate ion has a charge of -1. Chloride has a charge of -1. So that's the process by which you secrete H+ into the lumen of the stomach. So this acidic chyme within the stomach ends up traveling to the small intestine. You kind of have to neutralize this acid. It's not just going to neutralize itself. Otherwise, the amount of acid within the acidic chyme would be so intense that it would start digesting the cells of the small intestine. And so you need a method to actually neutralize it. And the way that we do that is by a secretion from the pancreas. And the pancreas can secrete bicarbonate ions, and it helps neutralize the acid. And so this particular bicarbonate comes from the pancreatic ductal cells. So you can see that there's the lumen of the pancreatic duct, and it's one of those cells in the lumen of the pancreatic duct that you get secretion of bicarbonate ions. And this process, just like what we saw in the last example, it requires a lot of carbonic anhydrase. And the carbonic anhydrase is very important in catalyzing this particular reaction. You're going to see this reaction dozens of times in this course. You're going to get really sick of this reaction, or you're just going to get so used to seeing it that it's going to become like second nature to you, that you know exactly what's going on. You know exactly what this reaction is. And so this reaction catalyzed by carbonic anhydrase is carbon dioxide plus water yields bicarbonate ion and a proton. And that's where our bicarbonate ions are going to come from. And so here is the process by which you get pancreatic bicarbonate secretion. So this is the ductal cell of the pancreas. And this is there's going to be a capillary on one side. The cells need oxygen, so these capillaries are going to supply oxygen, it's going to supply nutrients, it's going to take up carbon dioxide. And there's the luminal side of the, on the other side of the ductal cell. So the first thing that's going to happen is water, which is present in the cell anyways, and carbon dioxide, which is a byproduct of cellular respiration. That reaction is going to have carbonic anhydrase present, and it's going to create bicarbonate ions and H+ ions. The next thing that's going to happen is that bicarbonate ion is going to be transported across the ductal cell into the lumen of the pancreas, the pancreatic duct, so that it can travel to the small intestine. And it's going to be in exchange for a chloride anion. Again, electrical neutrality is maintained. One negative charge out, another negative charge in. The next thing that's going to happen is that you have to get rid of that H+ within the cell. That's dangerous to the cell. So just like that hydroxide ion that we saw, or anion that we saw in the parietal cell, the H+ ion, or the proton, if too many of those accumulate within the cell becomes extremely acidic. None of the proteins will work, none of the enzymes will work, and the cell will die. So the way that we deal with that H+ is that we get it out of the cell by using a proton sodium exchanger. So 1 proton out, 1 sodium in. Again, electrical neutrality is maintained. We're not going to talk about these transporters. This is really just to ensure that this transport process occurs. It sets up all the gradients that you need to make sure that the sodium proton exchanger is going to work. One thing we didn't talk about is this chloride anion channel. It's something called the CFTR channel. And so this is maintaining a concentration of chloride on the outside of the cell to help ensure that you can continually pump bicarbonate out. So 1 bicarbonate out, 1 chloride in, and that same chloride is going to now go back over to the other side so that it can help transport another bicarbonate. And this process is just going to loop. This CFTR protein is referred to as the cystic fibrosis transmembrane conductance regulator. Very important channel protein. It's extremely important in cystic fibrosis. In a person who has cystic fibrosis, this channel does not work. And the extent to which it doesn't work really depends on the specific genetic mutation that you have. Some mutations are worse than others. So some people can have a very mild form of cystic fibrosis. Other people can have a very severe form if the channel doesn't work at all. Normally what happens is that when you have this process working, with this particular ion channel working, normally where there's an ion that goes out of the cell, water will generally follow. And it keeps things well hydrated. This occurs in the pancreatic duct. It occurs in places like the lungs, the small airways of the lungs. You have this channel as well. And so if you don't have the ability to put chloride out, water can't follow, and you can't keep all your secretions very fluid. They become very thick. It clogs ducts. It doesn't allow things to flow. And the majority of people, when they think of cystic fibrosis, they think of the lungs, and they only think of the lungs. And that's true to an extent that there is a very serious respiratory effect of cystic fibrosis, whereby all the little airways get plugged with really thick mucus. But what people generally don't think about are the gastrointestinal effects of cystic fibrosis. And so the blockage of the pancreatic ducts. So you can imagine that if there's very thick mucus within the pancreatic ducts, it'll clog it, and any digestive enzymes that you'd like to get out into the small intestine can't get there. And that leads to a lot of problems. So that's a little bit on the cystic fibrosis transmembrane conductance regulator, the CFTR gene. This is the CFTR channel, and this is coded for by the CFTR gene. So speaking of mucus, mucus secretion occurs in quite a few areas in the GI tract. The most notable is in the salivary glands. The specific type of cells that produce saliva are called serous cells of the salivary glands. In the small intestine, and to an extent in the large intestine, in certain areas, mucus is secreted by goblet cells. And in the stomach, the specific type of cell that releases or that secretes mucus is called the mucus neck cells. And what mucus is really just a whole bunch of water with this thing called a glycoprotein. And these glycoproteins are a little bit of sugar and a little bit of protein. So carbohydrate plus protein. And the specific type of glycoprotein that comprises mucus is called mucin. People got a little bit, they got a little bit lazy with what they wanted to name things. And mucus is extremely important. It confers protection to the GI tract. So in the stomach, the acid can't digest or damage the cells of the stomach because there's a layer of mucus. In the small intestine, it again adds a little layer of protection. It also helps lubricate the gut so that food stuff can actually be transported across it with relative ease. The next type of secretion that you get in the gastrointestinal tract is bile. We're going to talk about bile when we talk about breakdown of fats. What bile is a really is comprised of a few things. It is a substance that is known to emulsify fats. It helps them become a little bit more soluble, and it's comprised of something called a bile salt. That's really the emulsification factor within bile. But there's also bile pigment, and this is pigment that is the result of breakdown of the hemoglobin within red blood cells. So if you've ever seen bile, it's kind of like a greenish color, and that greenish color actually comes from the broken down hemoglobin. It's sort of yellowy green. And then also within bile is a little bit of cholesterol. So that sterol-based compound that we talked about on day one. And it is secreted by specific cells within the liver, the main type of cell within the liver called a hepatocyte, or just a liver cell. Hepato means liver and cyte means cell, so liver cells. The bile doesn't generally go right into the gastrointestinal tract. It gets stored for a while, and it's stored in the gallbladder. And the gallbladder concentrates the bile. So it stores it and concentrates it about 10 times the strength that it was released from the liver. During a meal, the gallbladder will contract and it'll release bile into the small intestine so that it can immediately start to emulsify those fats into smaller droplets so that they can be managed and they can be broken down. So those smaller droplets together are going to have a greater surface area than a large fat globule. And so here's where we're going to get a little bit into the digestive enzyme secretion, because once we start getting into this region, this kind of bridges the gap between digestion and secretion. So digestive enzymes are secreted in a lot of places within the GI tract. We're going to talk about all of them. We're going to talk about the digestive enzymes of the exocrine pancreas, or the acinar cells, or those lobules within the pancreas. We're going to talk about the enzymatic secretion within the salivary glands. There's only a couple things that are secreted by salivary glands. We're going to talk about the digestive enzyme secretion within the stomach. There's a couple of them. And we're going to talk about the digestive enzyme secretion in the small intestine. There's a few of them. And so we'll talk about the specific enzymes in digestion. You'll find that the digestion section actually is a little bit more intuitive from a physiology perspective. It just seems to make sense with what the enzyme is doing and then what's happening. One thing that I want to leave you with is that when we talk about digestive enzymes being released, they are almost always released in an inactive form. And an inactive form enzyme is called a zymogen. So if you ever see that word, just means an inactive enzyme. And so these zymogens end up being activated in the lumen of the GI tract. The reason why it's very important for these enzymes to be released in inactive form is that it prevents damage to the cells that actually secrete them. So for example, you're going to have these zymogens secreted by, I don't know, a pancreatic cell. And that zymogen has to go through the pancreatic duct and then eventually travel to the duodenum, or the small intestine. Well, you can imagine what might happen if the pancreas released digestive enzymes in active form. As it's traveling along the duct of the pancreas, those enzymes are going to start breaking down all the proteins and all the lipids within the cells that line the pancreatic duct, and that wouldn't be good. So it's extremely important that you have these zymogens released in an area of the GI tract whereby it can become activated, but it's usually in an area of the GI tract that's lined with mucus. So it's lined with mucus, you release the zymogen, it sits on top of the mucus, and then you release other stuff that's going to activate that zymogen. So now you have an activated enzyme that isn't in contact with any of your cells. So I'm going to leave you with that for today. And next day, we'll talk about the process of digestion. We're going to talk about the chemical, we'll first talk about physical digestion, but then we're going to talk about chemical digestion from the standpoint of carbohydrates, lipids, carbohydrates, proteins, and lipids. We're actually going to combine the discussion on digestion and absorption, because it kind of makes sense to talk about both of them together. So our next discussion is going to be on the digestion and absorption of the various breakdown of your food stuff. I'll see you then.